Welcome, everyone. My today's guest is Sofija Vuckovic. She's a good friend, and she's also a PhD student at Karolinska Institute in Stockholm, working to investigate the role of smooth muscle cells in atherosclerosis. And she's also exploring their potential as a novel therapeutic target. Okay, so she's previously done her master's in innovative medicine. We actually studied together in this program. And this was a dual program where you can go to multiple universities. And her path was first year at University of Heidelberg and then University of Uppsala. She finished her master thesis in a completely different place in Hamburg, and we're going to talk about her applications to all of those, to programs, to master thesis, to her PhD, current PhD position, and all of that. Sofija, welcome to the show.
Thank you so much. It's a giant pleasure to talk to you here and on this platform about all these important topics. And I'm sure that I hope that some of the information that we discuss will be useful for people trying to do a PhD or doing a PhD already, or just curious about how the academic life is. So thank you for allowing me to be your guest today.
I have definitely allowed you to be my guest. It's a pleasure to catch up with you. It's been a while that we talked, and I'm sure
Exactly.
It's useful to some people aiming to apply to PhDs in Europe, and maybe just to get a glimpse of how it is to do a PhD at KI. So I gave a brief introduction of what you do and where you went to study for your master's, but of course you can tell me more about that. So can you maybe give me your brief academic background overview from your bachelor's, and how did you finish to do your master's? It's this Erasmus program and then getting to KI.
Sure, yeah. So originally I come from Serbia, and I moved from Belgrade, my hometown, when I was quite young. Now, when I look back, I was only 18, so I moved directly to do my bachelor's in the Netherlands, and I actually studied at Amsterdam University College, or AUC, which is an honors bachelor program that is managed by two different universities in Amsterdam, two big research universities, namely Vrije Universiteit in Amsterdam, and also the University of Amsterdam itself. So it's kind of like a smaller program where you are focusing on what you want, basically. So you have the fantastic opportunity to build your own curriculum and to get a degree in the end in liberal arts and sciences. And looking from this point, where I'm developing my career as a biomedical scientist, it's a bit weird to know that I have a bachelor's degree in liberal arts and sciences, but basically everything that I took in that program was a strong biomedical track. So I didn't wander around a lot. But what was really fun about this program is that it also had some mandatory classes that were in completely different fields. For example, some studies in anthropology, logic and philosophy, languages, even in literature. So it did give me a glimpse into some other things. So that kind of program was really fun. And there are several colleges in the Netherlands that give this kind of opportunity. So for people who might not know exactly what they want to study in the future, this is a good opportunity to really explore different fields. So that was maybe a bit of a long answer regarding my bachelor's, but then...
I do want to jump in there, sorry.
Yeah,
because
go ahead.
I will forget later. So you mentioned that you got this opportunity to build your curriculum a bit. You didn't wander too much, but you did get a little bit into philosophy, languages, and so on. Are there any of those subjects that gave you something useful for the science? You know, my background was completely science. There was no philosophy. There was no logic. That's a double meaning there. There was no logic. But I feel like I would benefit from some of those subjects. Like it would be cool to have had logic in my bachelor's. Were there any of those skills that you learned from those other not completely related to science, to at least biomedical science subjects that benefit you to this day? We'll be right back to the podcast, but first, I want to say thank you to our sponsor, Litmaps. If you are struggling with the academic literature and you are overwhelmed by the number of papers you are trying to read and make sense out of it, Litmaps is here to help. And visualize for literature, you can find research gaps, and you can create a beautiful map and keep yourself updated every time a new paper comes out that is connected to the map you've built. And if you're working on a review paper with other collaborators, this map that you build, you can also share with your collaborators and have them build it so that you can go back and forth in exploring the important papers of your field. Litmaps is a fantastic tool that I've been using for over 6 months now, and I've found it incredibly useful for many purposes, and it's completely free to use. If, however, you want to use the more powerful Pro version, you can use the code in the description, NN30, and you'll get 30% off on all of your first-time purchases with Litmaps. Thank you, and let's get back to the episode.
Definitely. I would say I would highlight two things. So the first thing is that the focus at AUC was really on learning how to work with people from different fields. So especially in this interdisciplinary environment where everybody has their own curriculum, basically, and no two students have the exact same track that they're following, you are forced to interact with people who have different perspectives, who come from different backgrounds. It was a highly international program also, which I think was quite useful for the future. And you are really forced to learn how to speak about your academic subject or interest in a way where everybody can understand it, even people who don't have maybe the necessary base knowledge to understand the nitty-gritty about it. So that was, I think, really important for later, for presentation skills, for talking to people who are not in your field necessarily, because as you know, biomedical scientists can be specialized in very different things, and there it's really cool to be able to talk to somebody who's studying, I don't know, diseases of the immune system, when you're somebody who is actually focusing on neuroscience. Of course, these two fields have some touching areas, but you're still focusing maybe on different cell types, different proteins. So that was a cool skill. And the other one that I would like to highlight is writing. AUC had a very strong focus on teaching students how to write. So we had two courses that were in writing. And this gave me a really strong foundation that I think got noticed also in my master's and in my PhD. The fact that I already knew how to compose review articles, the fact that I knew how to work with literature, how to collect my sources, and how to write coherent paragraphs. So I think this was something that was really, really useful and actually helped me land my first job. And I will tell you about that, I guess, in a bit. But those skills were really important. So maybe not what you would first think of, that, studying logic really strengthens your brain and your ability to think. That's definitely the case. But more than that, I would highlight these like more meta soft skills that were important for my career as a scientist.
I recently read this book called A PhD is Not Enough. It's quite a popular book on Amazon. It's quite a short book also. You can read it in a day or two. And it's this physicist who first wrote this book in the 90s, and there's a second edition from 2010. But initially, he's telling you how to survive in academia, right? And I think the majority of the book was focusing on telling you how important it is to have these soft skills, how to give a good talk, how to talk to other people, how to make your research proposal compelling and that other people can understand it, and all of these things. And giving a bunch of case studies on when that is not in place, how that can harm you in academia. So very cool that your university had this as a foundation skill. So you then went on to do this master's. We did together. I was very pleased to meet you there. How did you choose your master's? Did you have a big shortlisting Excel file, or what was your way of selecting master's program?
Yeah, good question. I think many people think, oh, it's just applying for universities, but especially when you're a student not coming from the EU and you're applying for the universities in Europe, it's important to figure out how to sort your applications, because you're probably applying for many. What I meant to say with applying from outside of the EU is that people often need scholarships. That was my first criteria, actually, when I was looking for programs. I knew that my family and myself couldn't really financially do studies abroad and masters abroad unless I got a scholarship. So what was attractive to me regarding the master's program that we did was the fact that you could get a scholarship. And I did get a scholarship in the end. So I was very, very lucky to be one of the students that had that opportunity. But I basically had an Excel sheet with all the different programs I liked. I needed the program to be in English, to give a scholarship, and to be, of course, interesting to be. It was mostly programs in biomedical sciences that were focused on research itself and that offered opportunities to be in the lab. And that's also one of the awesome things about our masters, that we basically had 1 semester that was courses and 3 semesters that was purely practical work in the lab, really putting you in the situation where you will be as a scientist and a PhD student later on. So I really like that. But I think in the end, I applied for only 5 master programs, which was very few compared to how many I applied for my bachelor's. So I didn't apply for too many, but the applications that I did send, I really made an effort to make them strong. And I think that was helpful, especially when it comes to getting a scholarship.
So those boxes to tick that you mentioned, like to have a scholarship, to be in English, to give some research opportunity, there's quite, well, I remember when I was searching to shortlist programs, there's not so many of them that tick all those boxes, because those were also my, criteria. Because I'm from Bosnia, you're from Serbia, we are not in the EU, and of course, you want to get an opportunity for a scholarship. Were those? So I want to ask you, in those couple of programs that you ended up saying, yeah, these are the good match. Were they all Erasmus programs, or were there some others? Just if somebody's looking to apply for those same criteria, what were those programs?
Well, I think only the one that we attended in the end was Erasmus. So I remember that another one, which I really loved, was in the Netherlands, in Utrecht. It was a program in regenerative medicine, but actually there I missed a deadline. So important message for all, take care of your deadlines and don't miss them, because it might cost you an opportunity. That one was really good. Then there was one that I really liked that was focusing on cardiovascular research, which is my particular field and my favorite field ever since I started learning about the human body. The cardiovascular research program in Vrije University in Amsterdam, but that one I couldn't apply to because they ended up giving scholarships only to people who did not live in the Netherlands beforehand. And I wasn't satisfying that criteria. So that program wasn't for me. And oh, that's a good question. I think one of the programs was also in Belgium. And
But the
I
scholarship
think that's all I can remember now.
itself was being given by the program, right? So that
Yes.
program, so that particular university, it's maybe worth going to each university website and trying to see if there are opportunities for scholarship for that program.
For sure, yeah.
Part of
How I
bigger funding scheme. Sorry.
Yeah, sorry to interrupt you. How I went about it is that I went to each university that I thought was strong and good, or that it happened to be in a city where I was considering living. And then I would search the programs. If there was a program in biomedical sciences, I would check the funding opportunities and the language of study and a bit more detail about the program. And if it seemed okay and it satisfied these criteria, then I would put it in my little Excel sheet. But it's really important to carefully study all the information that's on the website of the university itself.
I wanted to ask you, I did not tell you I'll ask you this, but I wanted to ask you, how was your interview for IMIM, for the international master that we ended up, studying together at? Mine was quite a disaster. I had,
Really?
yeah, I was just in I don't know, was it the same for everyone, but I was in between several exams. So it was like an exam week. So I had an exam the day before and I had an exam the day after. And I was completely sleep deprived for that whole week, as it goes. And I remember saying, I cursed something and I didn't even notice that I cursed. My screen was not, sharing for my presentation or something. And I don't remember this, but my mom, who was in the next room listening, she asked me later, why did you say bullshit in the middle of your interview? And then when I was loading the track in my mind, again, I realized When my thing wasn't sharing, I probably cursed, not even realizing that I'm cursing. How stressed I was.
Yeah.
So it was a complete disaster. How was it for you?
Wow, I didn't know that. That's a really interesting piece of information. And you actually did get a scholarship in the end and a chance to participate in this program. So I think that speaks of how strong a candidate you are and how good you are in these applications. But for me, I think it went quite well. I remember that I was in the building of AUC when I was doing it, and I practiced a lot for the presentation. So I was really ready. I was stressed about the time because we had, I think, a strict 5-minute limit, but it went well. I think why it went well was because I had another interview before that one, a couple of weeks before that, and it was actually in Groningen in the Netherlands. And it was one of the three programs that participate in IMIM, in our master's program. So if you remember and maybe for information for our listeners, the IMIM program, master program that we did is managed by 3 different universities: University of Groningen in the Netherlands, University of Heidelberg in Germany, and University of Uppsala in Sweden. And basically you can move through these universities in different ways, so that 1 year is in one of them and the second year is in another university. But the program in the University of Groningen that participates in this IMIM program was the master's program that I applied to as well. And I actually asked them if I can come to Groningen and do the interview in person. So I did. And I think that was really good practice. And that one was really scary. And I thought that the people who were listening to me, I don't know if they were having a bad day, but they seemed a little bit annoyed. I don't know if they were annoyed with me or annoyed in general, but they didn't give me a lot of positive signals. I did end up getting an offer from them and the scholarship as well. I didn't end up taking it. I went for the IMIM program, but I think the ability to practice an interview before the one that was most important for me was really helpful.
But what's the biggest thing you would recommend to someone who is supposed to have a master's interview that they should prepare or to practice, or what took the most time to practice for you?
Yeah, a good question. I think practice, 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 and I cannot say this enough. So if there is a task that you need to present something or that you need to have some slides, really polish it and practice your presentation so that you can deliver it in the allocated time, so you don't run over, and so that you are speaking coherently, because these are one of the most important criteria. But it's not that you're only presenting well, it's that you show that you can adhere to the specified time, so that shows that you can complete tasks and follow instructions. And another piece of advice: if your bachelor's university offers some kind of service to prepare for interviews or some study advisor that can help you practice, do that. Because I did that myself in Amsterdam, and that was really, really helpful, because you're talking to somebody you don't know. It's a little bit of a tense situation, but the stakes are basically non-existent. So you can really practice in an environment where it feels real, but it's actually not. So that's really important.
I'm smiling a little bit because you said, it's important to show you can follow instructions. And then immediately I go back to the time when I did that interview, and I definitely showed that I cannot follow instructions, because this task that we had to present a paper in 5 minutes, I went overboard. Even though I practiced the night before. That's what I tried. In the middle of these exams, it was just difficult to find the time in it. And also, the reason I was unprepared was I wasn't really expecting to get invited to the interview in the first place.
Right.
And I'll just let it be. I kind of shoot the shot and shot the shot. And I came not prepared enough. And I think it went to like 7 minutes, and then I abrupt. So it was just a complete disaster. I think what I can say as a positive tip, like how I managed to take out a positive response in the end, and what saved me in that interview, I think, was the Q&A afterwards,
Right.
where even though my presentation was not the best and it was not adhering to all of the instructions when it comes to time and all of that, I was really able to pick apart the paper, and I would be able to talk about that paper for an hour. And that's what actually ended up happening. This professor and I had a back and forth about the paper and about my plans and ambitions. The interview was supposed to be 45 minutes. I think we went double the time because we were
Oh,
just
wow.
chatting, and it was interesting. So, and I've heard this from several other professors, as a criterion by which they evaluate candidates. One of the professors said a very interesting sentence, highly established professor. He said, I'm looking for someone that I can chat science with. If we can go back and forth, if I can tell you an idea and you can understand it, you can grasp it, and then you can give me back something, and then I can, feed off of that and give it back to you, and we can just spend 2 hours that goes by fast in chatting about science. I think we are, that's the majority of the criterion that we can work together. Of course, there's other things, but for him, that's like a big green flag. So maybe
Yeah,
practice chatting about science, and about, if you have a project like your master's project, you could just try to talk about it to as many people as you can, and you will hear more questions about it. And then you will understand what people don't understand about your research, or what is the most interesting part about your research.
absolutely. And I would just add two more points from the story that you mentioned. The first one is start preparing early, because often these interviews come in very stressful times, like the exam period. And then, if you start preparing like 2 days before, then you're really stressed and you can't maybe be as thorough in this preparation and in this thinking about the paper, for example, if that's a task. You can't really invest in that task as much as it deserves. So that's number one. And the second point is that it's not a disaster, definitely, even if sometimes you feel that it is. And you can get an opportunity even though you don't expect it, because it's so fun for me to hear that you also didn't expect to be invited for the interview. I thought I would never get a spot in this program, and I thought I would never get a scholarship. But that's why it's important to be brave and go for applications, and send the email, send the application, do the interview, even though you don't feel perfectly confident, because you might end up getting it. And I think this is especially important for girls and women in sciences, because sometimes they can be less confident about their own abilities. So I would really encourage, especially everybody, but especially girls and women, to go for it, even though they don't feel like they might tick all the necessary boxes, because you might get it, because I did and you did. So I think that's an important point to highlight.
I think so too. It's a shame not to get an opportunity just because you self-disqualify, which is something
Exactly.
I guess women in science might tend to do more because of historic reasons and everything. Yeah, I would agree with that advice. Shoot the shot, even if it's feeling like you're not the perfect match for the program and you're not feeling super confident. Shoot the shot. I shot the shot, and the shot got fired, and good things happen. Okay, so then
Exactly.
you said this positive thing about this master's program was that you got to choose, and this is something I liked as well. You got to choose several different internships where you could just go into the lab, you get a supervisor, and you're free to do whatever you want. So we had people going to companies like Roche or AstraZeneca, or all these big pharmaceutical companies. You had people like me working in a fly lab, right? It's a basic science. You had people working something in between, something a little translational, but in academia. But then, how did you go about choosing those labs? And maybe tell me some of your experiences.
Yeah, I think that's a very, very important topic, because often I see students not knowing how to go about this process. And my first piece of advice there is just bother people. Just send them an email, pick up the phone and call them and say, hey, I think what you're doing is really cool. Is there any way that I can join your lab? So how I did it is that I searched the institutes and universities around, and even companies, and basically focused on cardiovascular sciences, because that was really what I was interested in. And for all of these different groups that I found, I looked if their websites or their some kind of page that they were managing had vacancies for master students. So if they had internship opportunities and projects already developed for somebody to come in and do. And if not, then in any case, I would just send them an email and say, I see that you're working on this topic. I'm really interested, and I think I could contribute in this way. And here's a little bit about my background. So basically I sent many different emails because very often you will get a no. Often you will get no response. And if you get no response, send a follow-up email. It's okay. You can bother people. They'll be fine. So that's basically how I did it. And in Germany, while I was doing my first internship in the second semester of my first year, I did an internship at the medical faculty in Mannheim of the University of Heidelberg, and I worked on cardiac fibrosis. And that was a really cool opportunity because I polished my skills in cell culture and in molecular biology. And that's another point. Find out what you want to get good at and pursue labs and groups where you can learn these things because sometimes you will get lucky, but you want to maximize your chances to develop your CV in the way that will serve your future plans, and not only the research group that you're in, but also focus on how you can contribute to the research group that you're in, because ultimately that will give you good recommendation letters. So there's many things to it, but basically my biggest encouragement would be to send emails to people and be proactive, because people like seeing that others, especially professionals in the field, are interested and want to join and want to hear about it more. And maybe the final point that I would say is be prepared for these conversations that you will have with these labs, because once you send an email, they will send an email back. They'll probably meet up for a little conversation about possibilities. Be prepared for that meeting, and that means read their papers and understand exactly what the research is. Prepare questions about the research, and prepare questions about what they expect from a student coming in, what their supervisory style is, how long the project is, what you will be able to learn, because that really gives you an opportunity to see if you're a good match. Because it's not only that you have to be a good applicant for the lab, but the lab also needs to be suitable for you. It's not only that you are reaching for the lab, but the lab also needs to be a good environment for you, because maybe you want on-hand supervision and the supervisor simply doesn't have time to provide somebody who's going to be with you in the lab all the time. And that is doesn't mean that you are doing something wrong, but that it's just not a good match. So be prepared for conversations with the labs.
This is 2 points. One, I love this that you mentioned this point about bothering people. I mean, in a way, you're bothering them by sending multiple follow-ups, but you're not really. You're just showing your interest. I had a couple of weeks ago a guest. His name is Ali Diwan, and he has a company called Trulitica, and he's helping PhDs transition into industry. He was telling me about his experience, about how he got his PhD. So his story is that he was in a completely different background. He was doing, I think, economics or business, and then he contracted Lyme disease, and he started reading about Lyme disease, got super interested in it, he recovered from it, but realized, we don't really know too much about the human body as I thought. I thought we got everything figured out, but we don't really know so much. I want to pursue this path. So he wanted to apply for a PhD in this lab that was studying Lyme disease, but he was late with the deadline. So his method was just, I'm going to bother this person until they understand I am the best person to be in this lab for the next 5-6 years. And long story short, after multiple emails and multiple conversations, they actually made a complete exception for him, even allowing him not to take a GRE, which was, I guess, a requirement at the time. He used his MCAT. So if you're really interested and passionate about a particular topic and what a lab does, being persistent can really help you. Okay, and then I always do this. I say 2 points and then I forget the second point, but never mind.
It's okay, I remember.
I wanted to ask you one thing. So there's many positive things about this program. One possible, well, not a negative, but a learning experience, and people can take it in different ways, which is that you move around a lot, right? So you had, especially your path was full of moving around. So there's Netherlands,
yeah, that's true.
there's Uppsala, there's Germany, so again, Germany in a different place, back to Sweden now for a PhD. How did, let's say if somebody is reluctant to go for a master's that has a lot of moving around, how would you help them demystify? How does this affect your mental health? Is this something really, really bad for you, or a really good learning experience? How would you describe that part?
My God, I would say it's both. It both sucks and it's amazing at the same time. So I think, first of all, I have to say that I'm really grateful for the opportunity to go through all of these different cities and compare in such a short period of my life. It was basically, 5 years, my bachelor's and my master's. I've been to 4 or 5 different cities in that period of time, and I think that's not that many people can do, and I'm really grateful. But at the same time, when it was over and when I was finally moving to Sweden for my PhD, I was so happy to know that I will just be in one city for the next 5 years and potentially, many more after that. So I was really happy for it to be over, but it was amazing because I could really compare and say, okay, I'm looking for a place and a city where I will have, these things that are more important for me personally. So it was that the city is pleasant to live in, that it's safe, and that people speak English, and that there are some international people around. Because, for example, in one of the cities I was in Germany, it was not a very international city, and I noticed that I didn't really like that and didn't always feel super welcome. Sorry, Germans. But that was my experience. And when I went to Hamburg, I was also struggling because I did not speak German, and I was staying in Hamburg for 6 months. I was not going to learn a whole new language for just 6 months, and can't learn German in 6 months. I think it's pretty difficult. So in that sense, I was really struggling. And in Hamburg, I didn't feel particularly safe either. So that was definitely not a city for me. But if I never went to Hamburg and had these 6 months that were kind of difficult, I would have never have known that. So living in Amsterdam also helped me identify, that Amsterdam for me is a great city to live in. And coming to Stockholm showed me that Stockholm is also pretty great. The winter is, hard because there's not a lot of sun, etc. But it's a good place to live in overall. So it was stressful to, ship boxes here and there, and then boxes would get lost, and you have no idea where you put, this T-shirt or this item that you're looking for, which box it's in. So it was pretty exhausting, but I think an exhilarating experience that enriched my life altogether. And in addition, made me grow up really, because these were my formative years from 18 to, what was it, 23, 24. And these most formative years, of your young adulthood, let's put it that way, to spend in this way accelerates your development as a person. It makes you more mature, makes you more appreciative of some things, clear about what you want and what you don't want. So I think overall it was a fantastic experience, but I'm happy to not be moving around anymore as much. So that's a long answer to the question. I hope it was helpful.
That's great. One of the biggest learning experiences for me, having the experience to travel around a lot for my master's, was that you really understand what is necessary luggage. So I learned that I really hate traveling with a bunch of luggage, and I ended up just giving up in the end. I went to US for my thesis with just a backpack. So I
remember that.
I'm just gonna buy a couple shirts there and whatever else I need, but I'm not dragging around and losing big bags anymore or breaking them in the air.
I never got good at that. I always have way too much stuff just dragging around left and right.
Yeah. Well, but it's good to settle down a bit. There is a time and place for everything, and I think, as you said, those formative years are the years to try something, try a bunch of things, and learn about yourself, what labs you like, what kind of supervision you need, what kind of cities you like, where would you like to be for the next 5-10 years, and then based on all of those inputs, you can kind of choose where to be long term, which is a great opportunity.
Yeah,
Okay.
that's
There's one thing I want to talk to you about, which is, I remember discussing with you. You were doing this master's. We were studying for exams, and at the same time, sometimes we would drink coffee, and you would say, "Oh, I need to run. I'm writing my review. I'm working on this review all the time, and I have some deadlines. I need to finish it." So you were right. I believe that paper is now published, right?
no, it's still not out. My
Still not.
God.
That's right. But it's kind of in the wrapping up stage, I guess. Can you share, as an early career scientist writing a review, how is that experience? Maybe some tips if somebody's embarking on that journey, and what to avoid, what tools to use overall.
Yeah. Prepare for another long answer because there's a lot of information here
Sure.
that I want to convey. Um, so this review was a part of that employment that I mentioned before, which I got, I think, in part because of my writing skills. So I was doing my bachelor's in Amsterdam, and one of the cool things that I did there was to have an opportunity to do a little internship for my bachelor thesis as well. However, I think I was doing the internship for like 4 weeks, and then COVID started. So my bachelor thesis was basically scrapped, and instead of doing lab work, I had to write a review. So my review was about hypertrophic cardiomyopathy, which is this genetic heart disease. And I wrote a little review, and it was in the end a nice bachelor thesis. But my supervisor at the time really wanted to continue this work, and she said, "Okay, I'm going to employ you part-time and remotely as some kind of research assistant." And I worked a couple of hours a week on this, developing this review into something more large scale. And by saying more large scale, we went really overboard, maybe, and we ended up doing a systematic literature review. And for those who are not familiar with the methodology, it's a very strict way of doing a literature review. You formulate a precise research question, and then you do a, a mining of several databases like PubMed, Web of Science, etc., with code, basically. So you input some code in these databases, and they spew out a bunch of different papers. Our research question was so large that we retrieved more than 5,000 articles. And then there's several steps that you follow in this methodology of the systematic literature review. And in the end, you're supposed to, remove papers that you don't need, and really, in the end, end up with a subset of the initial number of papers that you, in the end, read and extract data from. Because we started with about 5,200 papers, in the end, the papers we extracted information from were about 1,000, I think. And this is a lot. This is a very large systematic literature review. In hindsight, maybe it would have been good to have a more narrow question, but we really wanted to collect all the information in this particular field. So there's my first tip. Really be careful about what you want to do in the literature review. Define the question precisely and early. Invest a lot of time in doing this, because that will prevent you from 2 years down the line saying, "Oh my God, why was I so stupid that I, didn't think of this particular thing? Now I have to redo some things, and I've wasted a year investigating something that's not useful at the end." So that's the first thing. The second thing is be very familiar with the methodology, because people think, "Oh, literature review, I just Google some papers and that's it." But it's actually very beneficial for the strength of your paper to have a good methodology and a way to extract extract papers to construct your reviews. Third, it's really important to be familiar with all the tools that are available to people writing literature reviews. So these are both tools for managing your references, tools for citing while you write, and tools for systematic literature reviews, if that's what you're writing in particular. And I used Ryan, and it was pretty good, but others exist nowadays as well. So it's almost impossible to do it if you don't use all of these tools, because the numbers of papers that you're handling can be very high. So then it's really difficult to do it on your own. So using some of these tools makes it a lot easier.
Right.
So
What is this tool that you mentioned, and what is it exactly?
it's called Ryan, Rayyan,
Mhm.
and it's basically a tool that you can use to put all of these research papers in at every step of the way on the systematic literature review methodology. And you can include or exclude. You can have different inclusion and exclusion criteria. You can get all the information downloaded in an Excel sheet, which is really useful when you're submitting this paper and want to have a supplementary table. So it's basically everything you need to conduct a literature review in a systematic way, and you can add collaborators. You can see how much everybody has worked on it, the percentages of the papers that are labeled with certain tags, etc. So it's a really, really cool and free to use tool. At least it was when I was using it. But one more thing that I would like to highlight is that reviews take time, and they always take, as everything in science, they always take more time than you expected first. So I started working on this literature review in August 2017.
Wow.
So it's been, no, not 2017, sorry, 2020. I started in August 2020. Yeah, not 2017. But it's a long time. And at some point, the team of people working on this was 10. So it's a big effort to conduct a large-scale literature review, systematic literature review. It's similar, to doing a kind of like a meta-analysis, but from the literature perspective. So it takes time, and I think people need to realize that, because I, for example, had no idea. I thought this would be done in 6 months, and then it wasn't. So that's important to keep in mind.
And that's specifically because your lab chose to do this systematic thing with 5,000 articles. So
Yeah.
that increases the workload a lot. So
Yeah.
how do you, so let's say you started at, 2019, 2020, now it's 2024. It's still ongoing. It's maybe in the finishing, but it's a couple of years of effort to do something like this. How do you keep the organization there? Do you work every week on this review, or do you have pauses?
Yeah, that's a good point. I'm not on the project anymore. So since I started my PhD 1.5 years ago, I decided to step out and leave it to my colleagues in the lab in Amsterdam that's working on it. They were very competent and had quite a good way of working on it. But basically, people will come and go in science. It's always how it is. We will have students that are staying only for a couple of months. You will have PhDs that leave after 4 years. We will have postdocs that leave after a year or two. So it's important to keep the institutional memory, to document things very well, to describe why certain decisions have been made, and to keep all this documentation in one place, especially for such large-scale projects, to keep protocols, to keep email threads, and to have good handovers when people leave. It's always good to have somebody who is in charge of the organization, and in our case, it was a very, very capable postdoc who's also now left the lab. Actually, I know that she has moved on to another role. But she was really, really good at keeping all the documents in one place, communicating with everyone on why certain things were done and the way that they were done. And I think that really made the project possible, because otherwise I think we would have gotten lost somewhere on the way, and then it wouldn't be possible to get to where we were. And now I think the paper is close to publication, hopefully. So maybe in some time you will be able to read it. So let's hope that it will go well.
So I'm supposed to now write one review. It's not supposed to be a systematic thing, so it's going to be a smaller effort. But still, I do understand it will probably be more time consuming. Because I don't have the picture of how time consuming it is. I'm probably closer to thinking, this will be done in 6 months, but then it will probably take 2 years or more.
It depends on what you want to do and what methodology you want to use. But it's good to allocate a certain number of hours. I had it contractually organized that I was working this percentage of a work week. So I've always adhered to that. Sometimes you have to even put a bit more effort, but I had sort of designated times during the week when I was working on this during my master's. And afterwards, I had also stints where I was working on it full time when my contract changed at some point. So for me, that's how I organized myself. But in general, and I think this feeds into the conversation about PhD life in general. I think often you will be working on several projects at the same time, and then it's really important to understand the best way for you to divide your time across projects, depending on how much time they require and what you need to do, how urgent it is. and what works for you. For example, for me, it's best to work on one thing at a time. So to really devote one day or 1 week or 1 month on one project and not really, move between different projects in a work week, because I feel like that slows me down. But maybe some other people work in different ways. But that's why experience is needed. Over time, you will discover what in particular works for you regarding how to organize your time between different projects.
I think this gives us a nice switch to the next topic, which is the PhD life. So you said you started your PhD a year and a half ago, something like that.
Yeah.
So by now you had enough opportunity to have ups and downs. I guess lots of them. Can you tell me about your current progression of your PhD? What do you mostly work on, and what are the biggest lessons and the biggest ups and downs you can share with us?
Yeah. Wow. Where do I even start? So for me, doing a PhD is really fun. Maybe that's the first thing that I would like to mention. But in order for it to remain fun, I think you got to be careful not to overwork yourself, because I think many PhD students, work every weekend, work late at night, start early in the morning. I personally make an effort to not do that. So I can have, a rest. I can have time where my brain can just chill out, where I can give myself this so that during the day I can be creative and I can, think to the maximum capacity and really not make mistakes, because I notice when I'm tired, I make stupid mistakes during pipetting or something like that. So for me, that's really important. And the ups and downs. Well, I think 4 years is a long time of doing a PhD. In Sweden, it's 4 years, sometimes even longer than that, 4 or 5 years. And that will give you an opportunity to experience different phases in which it's stressful, phases in which you're intensely working on only one thing, phases in which you're traveling a lot for different courses, exchanges, um, um, conferences, etc. So I think my first maybe piece of advice is to not think that if a certain phase of the PhD, especially at the start, looks a certain way, that doesn't mean that it will stay like that for the entirety of the PhD. And I think that's a really good thing, actually. So regarding the ups, enjoy the ups, really, use them to the best of your advantage to accelerate your projects forward. But when it's down, know that it's not going to be, the down is not going to last forever. And the most important thing is to have grit and to really persist through these times and understand that it's a learning experience and that you're not supposed to be perfect at the stage of a PhD. You're still a PhD student, right? So you're still studying how to be a good scientist. That's what I would say. And get the support that you need from your environment. So at KI, we have to have something called a mentor. And this is somebody who's not in your team, but somebody who's external, and somebody who you can, call for advice or just to chat with about how things are going. And I think everybody, even though it's not formally appointed that you have to have a member, should have a member. Even at universities where that's not a thing, you can ask people, "Hey, I would like you to be my mentor. Can I call you every couple of months to just talk about my progression, my professional goals, my personal goals, even maybe, so that you can, have the support that you need." At KI, you can also have more than one supervisor. I have 3 supervisors, and that gives me a lot of opportunity to bug them about different things, because, one of the supervisors is my daily supervisor, and her I can ask if I, can't get something to work in the lab, or I'm just missing a reagent or something like that, really like basic daily stuff. I have my main supervisor who I bug for some daily stuff and some, large scale stuff. And I have a third supervisor who is the most senior person in our team. And of course, he's very busy, but he also has a lot of experience that's much more large scale and with a longer outlook. So he advises me on things like getting certain skills and having a larger overview on what I'm doing and why. So it's cool to have different people who you can bug for different things and get advice from. So does that answer your question? I said a lot, but I'm not sure if I answered.
I am a bit more curious if you could. I guess people have different ups and downs. For somebody, an up will be, a good feeling after a small experiment has succeeded. Somebody will get very neurotic about it. And, an up is not until I publish this amazing paper, everything until then is down. What are examples for you of what's a good day and what's a really bad day in the lab?
Yeah.
Specific examples.
Very, very poignant question. So I would say that for me, downs are periods. It could either be a day or even a month or a couple of months where I'm not focused the way that I would like to be. So when I feel like I'm wasting a lot of my time on tasks that don't amount to anything in the end, like if you're, reading a lot about, for example, a certain method, and then, a month later you understand that you can't even do the method because your lab doesn't have the instrument that you need. I'm just giving an example. So these kinds of periods for me really suck because they are not really rewarding. It doesn't amount to anything. You don't have a result that you can show at the end of it. So I really try to make every task that I'm doing worthwhile and that I make a little product from anything that I'm doing. If I'm reading a paper, make a little summary or note down what the paper is about so that, you don't have to read it again when you need it in 4 months. Or when I'm collecting some results, to collect them in a nice way, to, put it all in a couple of slides. Or when I'm writing a protocol, to really write it nicely so that in a year when I need it again, it's well documented and ready to use. So these kinds of things for me are satisfying. And if I have more of these sort of productive things, then for me, that's an up. I don't really associate it with, having a paper published or something like that, because that will happen at some point, hopefully. But it's more on a smaller scale that I look at certain ups and downs. So for me, it's really relating to focus. And if I can look back and say, okay, in the past 3 months, I learned this, I developed this skill, I managed to become independent in this technique or in this way of thinking, then it was an up. So it's a little bit in retrospect, but also how I feel at the moment. If I feel like I'm focused, if I feel like I'm productive, and that my mental energy is going through something that will have a result in the end, no matter what the result will be. It could be a couple of slides, but it is a result that I can, really show at the end of the day. And maybe a good thing to also mention as an example, during my first year of my PhD, we had a big delay. So one of our projects, my main project, was delayed by about 6 months because some experiments that we were doing didn't give us as good quality of samples as we wanted. So we needed to repeat a lot and basically pushed our entire timeline by 6 months because the person I was working on these experiments with went for a couple months of a leave because she just had a daughter at that time, etc. So things really, accumulated that made us push the project. And that really did suck because those results and these analyses were required for me to progress in the project and progress in my PhD in general. But I used these 6 months to go to conferences, to get to know the field more, get to know the experts, to do courses, and to learn techniques. And from one perspective, my focus was really scattered and I did many different things and didn't push any of my projects a lot forward. But I could look back at the end of the year and say, okay, but now I have so many credits from my courses that I'm basically done with courses until the end of my PhD. So that part, it was done. And I could also say, okay, I attended some really nice conferences and I have talked to these important people in the field, and now they know who I am, and hopefully they remember me and my poster or something like that, or at least a nice conversation we had. So yeah, it may seem at the moment that you're not doing something that's very worthwhile, but when you look back, it's important that you can look back and say, okay, these things I did. So with that in mind, it's good to always have a bit more of a large scale perspective why you're doing the things that you're doing. If you're doing an experiment tomorrow morning, why are you doing this experiment? On a larger scale, not because, so I can have 3 replicates in this particular method, but because I want to write this paper, and with this paper, I want to prove that this mechanism works in a bit of a different way than we imagine, or something like that. So it's important to have sort of a large scale on why everything you're doing in the week matters, and to keep that in mind. And of course, this is very easy for me to say. I don't adhere to it myself all the time, but I think that's what helps me feel motivated and productive.
I
I
really
hope that
love,
made sense.
yeah, of course, I really love this idea of productizing. Like we always talk about when you're trying to make yourself more productive, give yourself a reward. But that's kind of weird to reward yourself artificially. Like it's just weird. How am I supposed to reward give yourself a cookie? To me, that's kind of weird. But if you actually have a tangible, like a little product that you made, like for every situation, like a little protocol, a little couple of slides, or a slide with really organized results that are now ready to be used whenever you need them, to present to your supervisor, to someone new coming into the lab or whatever. If it's a protocol, you can take a look at it in 2 years, whatever it is, to make a little product. I really love that. I think I never looked at it that way on a more, daily schedule basis. I think I'll definitely apply that. I love that
Yeah,
advice. First
I
time.
owe that idea to my fantastic daily supervisor, Melody. So shout out to her.
That's really amazing. I love that. I think that's a very practical advice that I can implement. Okay, so I know that ever since you and I met each other, we were talking about this field. You're talking about the heart and how cardiomyocytes are the best cells in the world. You love the heart. You love what you do. But I never really understood what actually you're doing, and because I'm in a completely different field, completely different model. Drosophila doesn't even have a heart. So, you know, tell me what is it that you do? What are you trying to discover? What are your main? What is this large scale view of what you're doing?
Yeah, well, I do love cardiomyocytes, but I'm not working with them anymore. I have switched my favorite cell type to another muscle cell, and that is the smooth muscle cell. So I was working a lot with cardiomyocytes in my master's, and I think they were absolutely awesome and fascinating because they, have these spontaneous contractions, etc. They're super cool. For anyone who's curious about them, they can um, contact me, and I can rave about it more. But now I'm working on smooth muscle cells, and they're awesome for another reason, and this is their plasticity. So smooth muscle cells, for those who are maybe not familiar, are cells that can be found in various organs in the body, such as the gut, for example. But for me, they're important uh, when they are in the vascular system. So when you think of, the vascular system, one of the first things you might think of is blood pressure. Well, one of the cool functions of smooth muscle cells is that they're around the blood vessel, and they can contract and relax so that they modify the diameter of the vessel, and so they control the blood pressure. That's, for example, one really cool role of smooth muscle cells. But they're also very important for structural integrity of the vessel, and they're important for healing. So when there's a wound that harms the vasculature in some way, they can respond to heal that wound, and to reestablish the integrity of the blood vessel. However, that can also work in a sort of a harmful way. And an example of this is in atherosclerosis. And atherosclerosis is a very complex and chronic vascular condition that develops over several decades. And you might have heard of this disease where like fat builds up in your vessels. Well, that is atherosclerosis, except that it's not just fat building up. It's a very complicated procedure where it starts with some fat molecules going under the endothelial layers, and triggering inflammation, triggering this healing response from smooth muscle cells, triggering many different things. And over several decades, this wound grows, becomes more inflamed, more complicated, develops its own little blood vessels also inside this little lesion. And smooth muscle cells are really important during this disease because they kind of go crazy. They're like, oh my God, there's these many different signals. There's an injury, but it's not really an injury. And there's a lot of cells dying and there's these fat molecules. Oh, maybe I should eat them up, or maybe I should produce more extracellular matrix to make this endothelial layer more stable, etc. So the smooth muscle cells go kind of crazy and they change their phenotype completely, because normally a muscle cell, which is what they are, is a cell that contracts and relaxes. But in this particular disease, they can become a cell that resembles fibroblasts, for example, or resembles osteochondrocytes, which we associate with bone and calcification. They can become cells that produce a lot of immune modulators, so we associate it with some immune functions. And all of these processes are important for the development of the disease, and at the end, they can have a consequence for the patient. And maybe this is already way too much of a story and too complicated for people to understand. But the most important thing is that if these smooth muscle cells behave in a more beneficial way, the patient is less likely to have a harmful consequence, such as a heart attack or a stroke. But if the smooth muscle cells are more harmful, more destabilizing for the entire little wound and lesion that's happening, the likelihood of a heart attack and stroke is higher. So if we learn how these smooth muscle cells change their profile, change their function, and if we can learn how to impact that, we can potentially have a whole new vascular therapy. And that's really exciting.
You mentioned that when you have this plaque and the lesions that are piling up in our blood vessels, I guess, smooth muscle cells can take over a different kind of phenotype. They can become more like different types of cells. Is that usually a mesh that all of the smooth muscle cells that are there are having just this large mixture of differently behaving smooth muscle cells? Or is it different from plaque to plaque? So maybe my atherosclerosis will be one where my smooth muscle cells will be taking more of this bony phenotype, whereas yours might be a little bit different. What does this depend on?
That's the topic of my PhD, so I don't know. Is a short answer to that. But we expect the lesions to be very different from person to person, and even within a certain patient. If they have a lesion in this particular artery, the carotid artery on one side, it could be different on the other side. So it depends on the local cues and also the cues in the entire body. It's of course different in relation to genetics. It's different in relation to lifestyle. So this question is really complicated, and the smooth muscle cells in a particular lesion in a particular patient will be of various different kinds. They might be more of this type compared to this type, but usually we'll see all of these or many of these different phenotypes in one particular lesion. What is important to mention is that it's not like it's a switch from 0 to 1, but it's a whole spectrum of different functions and different genes and markers that these cells express. So it's hard to say, okay, this one is exactly the bony type and this one is exactly like a fibroblast. It's more complicated than that. But it definitely definitely differs from person to person, and this we're trying to figure out what it depends on and if we can use some kind of therapy to improve the clinical picture.
There is I remember from I went to a medical high school, right? So we had some subjects related to pharmacy, and I remember one of our professors mentioning about atherosclerosis, and she said that these little plaques in the blood vessel, they start accumulating already, you think you're young, but already at age 18, 19, it's starting to build up. So I remember a question that I had in my mind at the time, which is, does this accumulate regardless of my lifestyle? Am I just doomed at some point to have these plaques? I guess it's heavily dependent how much it accumulates. And then, is it reversible? Can I just, with my change of lifestyle, get rid of these plaques? Or is it something, once it builds up, you can maybe a little modify, but you don't have too much influence? You need the therapy.
Yes. Well, I think that everybody is doomed and that everybody has this, especially with how we live at the moment. So we even see some changes at the vasculature at these um, positions where these lesions usually develop already in babies. So this doesn't necessarily have to only be influenced by lifestyle, but maybe it's just a human condition that the way the blood flows in certain areas and this turbulent flow may already start up some inflammatory processes that lead to some changes. However, it's important to note that the fact that you have these changes, and even if you have an advanced lesion, it doesn't mean that you will suffer from a heart attack or a stroke or some other consequences. You might, never feel that you have these lesions and eventually, die with them instead of from them. But what we do know is that atherosclerosis is the biggest killer on the planet. And people usually think, oh, it's cancer or, I don't know, it's some other disease, but it is atherosclerosis. And until some of the recent data indicates that it kills 1/3 of all, the 1/3 of all deaths are related to atherosclerosis.
Okay.
So it is a big proportion of the premature death and the disability that we see nowadays, and it's a huge cost also for the health systems, etc. So the question is it reversible? Well, right now we don't have anything on the market that really reverses the disease. It only treats the symptoms. So we have these, balloons and stents that can sort of push the lesion, and open the blood vessel. But that's also problematic for some reason, because that itself can have consequences if there's like a thrombosis happening, or if the plaque just comes back, because that can also happen. So right now we don't have sort of a precise therapy that can reverse the plaque, but some new cool research, this is very recent stuff, indicates that in mice there are certain ways to reverse development. In humans, that has not been documented as much, but the hope is that smooth muscle cells are one of these targets that we can use to have this reversal, maybe even, or at least not making it worse, because this disease gets worse and worse over time. If we can at least stop it at some point, you can have it. You can have these lesions, and you can have even many different lesions, but in the end, they might not have any symptoms or any consequence.
Yeah. I wonder those babies that get get detected, those plaques get detected in those babies. I wonder if the lifestyle of the parents is something that could influence that. I guess some people do.
Very good question. Yeah, lifestyle of the parents, conditions in utero, and genetics could all be things that lead to these changes. Absolutely.
Awesome. Can you still see me? I seem to be frozen a little bit. You can see me?
I can see you, but you are a little bit frozen.
All right. So I think multiple technical issues are happening. So I think I might stop moving so that, because I'm frozen or mildly frozen. Okay, I'm back. So I'm going to cut this part out. Okay, so what could I finish with? So the most promising targets, or I guess the most promising new research for atherosclerosis is targeting these different, maybe reversing these phenotypes of smooth muscle cells. What is something exciting that recently came out, maybe that blew your mind in your field of research, that you said, oh my God, this is amazing. What a paper. This is a new route. So there's obviously your topic. What are some other sub niches of your research that people are doing cool stuff in?
Yes. Well, there's a lot, I have to tell you. There's a lot of research in this field, and papers are coming out all the time about smooth muscle cells and other cells involved that really make me rethink how this disease works. And maybe one thing that continuously blows my mind is how complicated this disease is. And that really makes it hard to do any kind of translation into the clinic and translation into the human system, because a lot of the research is still happening on animals. So I would say that is one thing that really blows my mind, that I'm constantly fascinated how the human body is so cool and so complicated, and how much we don't know. But maybe one thing that I could highlight is not directly related to smooth muscle cells, but it's related to the interaction between the nervous system and inflammation. And there was some lecture I attended last year, or a couple of months ago, I don't remember, where the group showed that if you stimulate the vagus nerve, you can, what was it, improve your condition of your vessels.
Right.
So through inflammation, so that the nervous system has certain anti-inflammatory cues that in certain conditions it can give off.
Right.
So maybe you understand this more than I do, because you definitely know more about neuroscience than me. But basically, this unexpected, or maybe for me unexpected, relation in between different systems in the body is something that fascinates me. And maybe that's a good way to, link it all the way to the beginning of our conversation, where I talked about liberal arts and sciences and the importance of knowing how to talk to people from other fields. I think in biomedical sciences, we can very often be focused, on one cell type, one protein, one cascade. However, we need to keep in mind that our bodies are so complex and so big and so interdependent. The systems in it are so interdependent that we have to be prepared to have a bit of a wider view and talk to experts from other fields, because there might be more to discover than it seems at first. So we should keep that in mind.
I love what my master thesis supervisor, Dragana, likes to say. It all has to connect in the end, because it is one system. We were having this discussion about one project that I want to do, and she was generating all these different parts. I want to shut down, genes in the nervous system to see how it connects to inflammation and how it might control the immune system in general. And she was asking these questions, how does a larva of a fly know that she's on her back versus on her front? Like, she has to sense it somehow, and that sends some kind of information to move back and forth. How does she smell food? That has to be connected with her motor system somehow. And then she has to be able to detect harmful substances. And then, maybe there are these phenotypes where with a little simple larva will roll. It all has to connect in this one large system. So I like that sentence a lot as a message. Like, it all has to connect in the end.
Yeah, it's beautiful.
So you mentioned the vagus nerve. This is quite an interesting topic of subject. Recently, I also saw one paper, and just recently came. I don't know if it's still even peer reviewed. It was like an accelerated Nature publication. I think it's titled something like a brain-body axis that controls the immune system, where they basically injected the mouse with LPS, so to inflame the mouse, but not to have any particular pathogen and multiplying. And then they recorded the brain, and they found these neurons that are reacting to something. So not really sure in the beginning what they were reacting to, but they were reacting to something. And then they find that actually vagus nerve can sense the accumulating immune cells, and that sends the signal to these particular neurons in the brain that say to the immune system, hey, maybe tone down the production of immune cells. And then they play with mice genetics, and they silence these neurons. So if you silence these neurons, the inflammation goes up way too much. And if you activate them, you suppress. So it's basically like a immune center in the brain that's telling the rest of the body, hey, tone down. It's nothing to do with the pathogen itself, but just sensing the amount of accumulating immune cells. So I think this is quite an interesting field. I'm not sure, it's still, time will show, and reviews will show how much of it we actually know, and we'll know more about it, but it's definitely fascinating. Sofija, thank you so much for talking to me today. It's been a pleasure.
Thank you.
It
was very fun.
Yeah. What are your plans for the next couple of months, couple of semesters for your PhD?
Yeah, the next task on my to-do list is to learn bioinformatics, which is a huge task, and I'm not, of course, planning to learn everything, because it's a whole field of study for itself, and I'm just a humble biologist. But I have to develop some skills, some elementary skills in bioinformatics, and I think everybody going into the field of biomedical sciences nowadays has to do that, because it's very big data that we're working with all these new technologies such as proteomics, transcriptomics, spatial. approaches. We really need to be good at analyzing the data that we get from these experiments. So that's on my to-do list. And other than that is polish further my ability to focus and to work on one thing at a time. And this is probably something that I will be working on for the next 2 years until I finish my PhD. So that's the idea. Yeah.
That's awesome. Good luck. I hope this is just the first of many times that we talk in a recorded session. We'll talk, of course,
I hope so too.
much more unofficially, but I hope we talk again on a podcast. I actually will try to implement your habit of productizing like every little task. So I think that'll be very useful. Thank you very much, Sofija. Till next time.
Thank you. Till next time.